はい、皆様、こんにちは。今日はですね、リンク J のライブウェビナーにご参加いただきまして誠にありがとうございます。日英ライフサイエンスセミナー Vol.2、英国がグローバルをリードするライフサイエンス系事業紹介、ということでですね、具体的には AI 創薬、プロセス最適化編をオンライン形式にて今日はお送りします。私はですね、リンク J の常務理事をやっております、曽山と申します。どうぞよろしくお願いいたします。今日はですね、画面でお示ししているプログラムに沿って、進めてまいります。皆様からのご質問、ぜひお寄せください。ウェビナーの Q&A 機能からお願いいたします。パネルディスカッション後半ですね、この時間にお答えをしてまいります。議論の盛り上がりとかですね、質問数に応じて終了時間が10分ほど延長する可能性がございますが、何卒よろしくお願いいたします。またですね、お願いが2つございます。ウェビナー終了後にアンケートフォーム、が表示されますので、ぜひですね、今日のイベントのご感想をお寄せいただきたく、よろしくお願いします。また、もう一つ、ウェビナー中の録音、録画、画面のキャプチャーなどはお控えいただきたく、よろしくお願いいたします。このイベントはですね、英語字幕ご利用いただけます。字幕はですね、市販のアプリを使用しておりますので、その内容についてはですね、リンク J、オクセンティア、バイオスパイア株式会社、特に誤訳とか訳文の欠落等ですね、責任は負いかねますので、よろしくお願いいたします。このシリーズはですね、オクセンティアのシニアコンサルタントであり、リンク J サポーターでもある松永正史さん、にモデレーションをお願いしております。松永さんはですね、もう長年にわたって、リンク J の、英国における、また欧州における顔としてですね、リンク J をとてもですね、サポートして一緒にいろいろとやらせていただいてからいる方です。はい、それではですね、松永さん、よろしくお願いします。す
すみません、佐山さん、過分なご紹介いただきまして。松永と申します。在英、今、オックスフォードにいるんですけれども、在英サポーターとしていろいろ頑張っております。今回、3回目になるんですけれども、オルガノイド、それから細胞治療編ということでやらせていただくんですけれども、10月のですね、11日にはですね、フィジカルで、英国ライフサイエンスシンポジウムというのが、開催されます。で、8期間、フィジカルで、ビザが緩和されるギリギリでみんなビザ取ってるんですけれども、行きます。今、結構日英が熱いかなと、実証してるんですけれども、いろんな形でネットワークを作っていきたいなというふうに思っております。よろしくお願いします。じゃあ、早速行きたいと思います。So, if I can introduce first speaker, Josefa, Dr. Josefa Amichis. He's head of the business development of Celes Limited. And this company is quite focusing on the organoid, and the organoid is quite getting famous. However, it's quite difficult doing the quality control or scale up, etc. Even though there's many academic paper we can see online or in the field. And Josefa has got experience in the life science sectors, even both for R&D and CRO industry. So he is now in touch as like an interface between the customer plus internal scientists, if I'm correct, Josefa. Yes, so all for you, Josefa, for you got 20 minutes talk.
Thank you, Meshashi. Thank you, everyone, and thank you for the opportunity to discuss Celes on this Link J event. Let me share my screen. Is that working fine?
Yes. Yes, we can.
Okay, cool. So, yeah, I'm going to chat and discuss with you about the company I work for, Celes, and the topic of this chat is how we are trying to enable the use of patient-centric models, in this case, patient-derived organoids, to revolutionize drug discovery and development. And we have a top line overview animation of our sort of business plans and our aims and the use of organoids on our website. So you can see a YouTube link there if you would like to see that. So, so who are we? So we're a sort of a hybrid biotech company combining bioengineering and stem cell biology, and we're basically a spin out of 2 UK universities that are combining the knowledge of biology, bioprocessing and stem cell knowledge. Our focus is developing the industrial manufacturing processes for organoid production at scale and ensuring that those organoids that are being produced are high quality, quality assured, and fit for purpose for industrial scale use. And these are high throughput applications such as high throughput screening, phenotypic assays, functional genomic screening such as CRISPR, and providing biology for on-the-chip applications such as and microfluidics. We believe that organoids are superior models of biology since they are derived directly from patients themselves, and organoids themselves are stem cell derived models. They represent the origins, the molecular features, the genomics, the morphology of the original patient tissue. So we believe these are attractive model systems that basically combine patient material with the scalability of in vitro cellular models. So how we do this, we have a patented novel technology which combines bioprocessing and unique bioreactors to basically scale up the production of organoids to scales that cannot be achieved manually. And then furthermore, we have stringent quality control, quality assurance, and a quality management system to ensure what we are manufacturing is high quality, fit for purpose for the applications they're going to be used by our customers. And I'll move on. And on the picture on the right is of one of our colorectal cancer models, which has been visualized using the Yokogawa instruments, and then basically they've been pseudo colored by the different Z stack images that have been taken. So as I mentioned, we believe that patient-derived organoids or PDOs are superior in vitro models. They're multicellular, they're 3D, and they can be generated from the vast range of epithelial-based solid tissues such as gastrointestinal, breast, liver, prostate, and there's plenty of papers out there that now show that organoids that have been generated and tested against compounds, they show a predictive and faithful correlation between the organoids and the patient themselves. And this has opened the doors to personalized medicine, which has been taken off with the likes of Senjin and Tempus. These patient-derived organoids, unlike 2D cell lines, are genetically diverse. Each has been generated from a distinct donor, so you have different demographics, different morphologies, different resistance or susceptibility to drugs, and different mutational profiles. As well as this, organoids, though they are generated from epithelial adult stem cells, they can be leveraged with other cells such as vasculature, immune cells, stroma, to create more complex in vitro models, which are sought after and have always been sought after as alternatives to animal models, especially now that immuno-oncology programs are starting to accelerate and there are no clear models on how to test those assets. What Celeste introduces and enables is a scalable bioprocess using unique bioreactors that basically enables large-scale batches of organoids to be manufactured to be used in high throughput applications. Above this is this quality assurance that those organoids, those batches of organoids, are tested with various different QC suites of packages to ensure that they are fit for purpose to be delivered into the labs of our customers, so health and safety kind of elements. Perhaps we check for genomic stability, we check for disease type organoids, we'll test against known compounds to ensure we have our process hasn't introduced any kind of genetic aberrations, or we haven't introduced any resistance or a susceptibility to known compounds. Celeste, our dedication is to the manufacturing, the industrial processes for producing organoids at scale. So it's the scale up and the quality control. The end use of these high quality research tools we leave to our customers and our collaborators in whichever applications they might be. Our focus is on growing these organoids at scale to qualities required for pharma and biotechs. We also have an element where we are kind of generating organoids as an off-the-shelf product. So in essence, we are producing these organoids at scale and then vialing them in a cryopreserved format with distinct SOPs. So our customers can purchase these organoids and in essence, basically for them, plate them, run their experiments and generate nice proof of concept data on patient-derived material. We want to enable the use of organoids earlier in drug discovery and make it easy to use. So I touched on this already. So the organoid market development is everly growing and accelerating. And where we see Celeste and what our USP offers is applications that require high quality, high reproducible organoids at large scale, whether that's millions of organoids or even tens of millions of organoids. And where these applications include is high throughput screening or phenotypic screens, CRISPR edited organoids to find and discover novel druggable targets, leveraging these multicellular models with other cell types to create complex in vitro models, and furthermore supplying biology, patient-derived biology for organ-on-a-chip systems for toxicology applications. So that's where our market pool is right now for large-scale batches of high-quality organoids. As I've sort of touched on, plenty of papers out there now are sort of showing the advantages of organoids. This is paper from 2018 that kind of shows the predictive nature of organoids, linking compound response to actually what was actually seen in that patient where those organoids were derived from. And this opened the door for personalized medicine. And only recently now we're seeing the use of screening organoids to develop clinical candidates. And this is, I believe the first clinical candidate using organoids and high content screening for colorectal cancer. So, and as pointed out in my text, they were able to show that they could develop a lead agent in 5 years. So condensing the timelines in drug discovery obviously has a dramatic effect on the cost of drug discovery programs. So this again shows evidence of using patient-derived models earlier in drug discovery. So at the moment, organoids are usually grown manually. This is obviously time consuming. If you were to scale this up to produce millions of organoids, you would need lots of people, lots of hoods, lots of plates, and perhaps using lots of labs around the world. And as you can imagine, this would basically bring batch to batch variation, lab to lab variation, and it's not a scalable process. So how do we bring large scale batches of organoids earlier into drug discovery? So this is where we've come in with our USP. We have a semi-automated process, which basically using our bioreactors, using pumps, inline monitoring, provides a consistent environment in our bioreactors for these organoids to grow, to be harvested as large-scale batches that can be delivered to customers. Right now, we have a first-generation bioreactor, which we believe is increasing productivity of organoids by 20-fold, and we have in development a second generation of bioreactors where we hope to be able to deliver 20 to 25 million organoids in one batch in one go. So here's an example of our first generation fed plate bioreactor. So in essence, we are growing these organoids, and we term organoids as those that are adult stem cell derived that are grown in a solid hydrogel. So the organoids are seeded in a matrix layer at the bottom of a dish. We provide a continuous perfusion of media, which basically provides this consistent environment for the organoids to grow, and in essence allows us to harvest a consistent and reproducible organoid final product. We monitor the media for waste and other metabolites such as lactate, glucose, and others to ensure that the environment they are growing in is exactly what they require for this organoids. And then at the end, we harvest these organoids, and we can harvest batches of 4 to 6 million organoids with this current setup. I'll move on. And so here's an example of showing this kind of like the what's going on right now. So to perhaps grow 5 million colorectal cancer organoids, if you were to do this manually using static culture techniques, using domes, you would need 45 plates. Now you can imagine how much work there would be required to grow this many organoids with this many plates, hoods, incubators, whereas we would basically use our first generation bioreactors in parallel with one operator observing this whole setup. There's pumps and sensors and monitoring of this system to ensure that we're basically providing this environment for these organoids to grow and to be able to harvest a large scale batch. Our second generation, as I mentioned, is more akin to chromatography columns. So we're aiming to achieve a fully scalable process, whether it's small scale to large scale, depending on what our customer requires. In essence, what we're trying to do with this whole bioprocess and bioreactors is to reduce handling time, standardize protocols, and in essence, give better, precise culturing conditions that ultimately leads to reduced batch-to-batch variation, improved yield, and also improvements in the size of the organoids that are harvested, as there is a correlation between the size of an organoid and its viability. So if we can produce a nice standardized size of organoids to our customers, they will get good data from when they use it in their applications. So as I sort of touched on, our sort of main USP using our bioreactors and our bioprocess is to be as a CDMO. In essence, we take existing organoid lines and we scale these up for our customers into either large scale batches as a one-off, or what we are looking for are for large scale batches over a regular occurrence. So this could be expansion of our customers' own organoids that they've either generated in-house or they've accessed through a biobank such as the Hub or ATCC or other biobanks around the world. They transfer them into Celeste and we develop these, we scale these up, and then we deliver large-scale batches over a once or a regular occasion. We can also expand the organoids that we are bringing into our own portfolio, which I'll touch on in a minute, and deliver these back to and scale these up for our customers. We're already supplying some of these, our colorectal lines, as an OEM product to Merck Millipore, and it's on their catalog. Furthermore, as a more bespoke nature, we can look to source organoid lines around the world, or even generate them directly from human biopsy tissue or from PDX tissue, and then develop these, scale these up, and deliver back to our customers. That's our main program. Our other program is to, in essence, offer organoids as an off-the-shelf format to distribution partners or directly to customers. So right now we have 10 colorectal cancer lines and we have 3 breast cancer lines, which we offer in this assay ready format. So our customers can purchase these organoids from us. They take these cryopreserved vials, they can thaw them, they can plate them or put them on their chips and run their experiments. And we're delivering in these vials enough organoids to, in essence, to maybe 3 to 5 384 well plates worth of data. So you know, I call these enablement products to basically get our customers some really good quality proof of concept data using organoids, make it easy for them to use organoids in their research programs, which would hopefully lead to them coming back to Celeste for large scale batches of organoids, whichever what fits their programs. We are now looking to develop, based on the market pool that we are seeing from the pharma and biotechs, normal colon and normal duodenal lines. So these are healthy lines. So these are for more toxicology type applications, such as gut barrier integrity. And furthermore, we have in development pancreatic cancer and esophagus cancer lines. And then with our customers, they are transferring lines from a wide range of tissues, both normal and cancer, such as non-small cell lung cancer, such as esophagus cancer, and such as normal colon. So we're doing a vast range of programs that are basically also validating our platform that they can scale up different tissues, different diseases, and scale these organoids at scale for our customers. Some data, I think it's always good to show some data. I talk about reproducibility and consistency of our large-scale batches of organoids. So we commissioned a program with a partner where we sent a lot of our different lines of colorectal lines and asked them to test against some known compounds against colorectal cancer. And this data in essence shows the high quality of the batches that we're producing. You can see this ISO 38 line was tested against a tyrosine kinase inhibitor, WEE1 inhibitor called adavosertib, and you can see the really nice dose response profiles for that compound on 6 different plates which these organoids were plated on. And I think the key metrics that the researchers that are doing screening type experiments are looking at are Z prime values. So you can see this is above I believe it's above 0.6, 0.5. So these are the key metrics that screening operators use for establishing that they've got a valid test to test organoids. And so we're showing that we can produce good quality organoids for screening type apparatus. Also, I think what's vitally important is organoids used in other outputs where perhaps we can look at the subtle effects of compounds on 3D multicellular structures. So again, adavosertib used on this line showed a nice dose response when we were using such as a phenotypic readout such as PDO counts or PDO volume. And what we see here is we can see more mechanism of action of this compound against the organoids. In this case, the compound maintained a small cores of viable cells in response to adavosertib. So again, what we want to show is that we can manufacture high scale volumes of quality assured organoids, and that these can be used to create good value data for our customers and collaborators. And lastly, I'll just finish off with our 4 main business drivers at Celeste. So the first 2 is what we do internally at Celeste. So we're continuously developing the scale up, the bioprocessing, and also the quality systems, the quality management system to ensure that we can scale up organoids and that they are high quality. We're continuously looking to expand the portfolio of organoids that we have in our sort of bank, just to show that our platform is valid for different types of tissues, normal and disease. And then the 3 and 4 is really what we do with our collaborators. I think this is where we can supply high quality organoids, and that these organoids can be used to create good quality data to further enable the use of organoids. So whether that's functional analysis or morphometry, or whether that's creating new complex in vitro assays. So really, we're looking to build relationships across the organoid ecosystem to further drive the use of good patient-derived models earlier in drug discovery, as I've listed on this bottom. And I think I will stop there, if that's okay.
Thanks very much, Jose, for a very nice presentation. And thank you very much for keeping on time.
Oh, thank you. I didn't know what the time was, so thank you.
No, because I used to work for another stem cell company and having organoids, and the experience quite difficult to control the size and the quality, function, etc. Organoid is quite like nasty, with having like a 20 micron to big one like a 600 microns. So if you can control that manner, you can help many biotechs, plus maybe academia. They are good in producing organoids. Scale up is maybe another business. So maybe
you're absolutely right. So where our bioprocess and our bioreactors, we do not change the way these organoids grow. So if an organoid grows very slowly, our bioprocess doesn't speed this up. We do not want to change the fundamentals of this biology that's been derived from patient material. So what we're introducing is this scale up to provide some longevity and more value to a patient material that's been kindly donated. Being able to offer this to pharma and biotech so they can basically use better models earlier in their drug discovery.
Thanks very much. And for audiences, if you have questions, please put in your Q&A, either in English or Japanese. English is preferred, but I try to translate. Right, good. Thanks. Josefa, don't leave. You must stay for the panel discussion. Thanks very much. And the second topic, first of all, I have to apologize, but the second speaker, Marcus Yao, he's a good friend of mine for another 18 years. He has been in trouble in flight cancellation from his European trip last night. And he is technically on the 30,000 feet at this moment. So I asked him last night, like midnight, to prepare like a 20 minutes good presentation. So video is prepared, and he is now trying to set up new businesses for the liver cell therapies. So Marcus has got experience for the iPS-derived company. However, this was an in vitro situation. And he did as a CEO of Definigen for 8 years and learned for the further processes suitable for cell therapy applications. So cell therapy application needing like a much larger scale of cell production and of course stability and how to transfer to the liver in vivo. So video is prepared, and if LinkJ team can start the presentation, please don't leave because we have panel discussion later.
Everyone. Firstly, I would like to Good morning, everyone. Firstly, I would like to thank Masashi at Biospire and J-Link for arranging this seminar. I'm going to start by telling you a little bit about the company. Aculiv Therapeutics are a Cambridge, UK company which specialises in the development of transformative cell therapies for liver disease. And the company technology licensed from the University of Cambridge. This technology is transformational. It enables the production of cell therapies from stem cells with a functionality and a scale that's optimized for liver therapeutics. So it really is going to transform the field, one would hope. This particular development has been built on years of stem cell creation, somewhat an older technology which takes stem cells and by introducing growth factors in a systematic way directs those stem cells along a given developmental path to produce liver cells. This older technology utilized directed differentiation, and the resulting cells had what you might call fetal functional adult liver cells, so they couldn't quite undertake the detoxification functions which are the primary role of the liver. Aculiv Therapeutics is the advance. It's developed a methodology to overcome this limitation and produce very, very high quality functional liver cells with the associated tox functions that are necessary for adult development and maintenance. So this slide overviews liver disease progression, and it really shows that the liver as an organ is the major detoxification mechanism for the human body. And in terms of the causation of disease, this can come from many sources. It could come from your genetics. There's a new disease called PNPLA3, which is going to be, and is actually, the focus of intensive pharmaceutical development activity, because it leads to the buildup of fat around the liver and ultimately leads to cirrhosis and scarring. Also, your lifestyle can play a huge part in disease progression. Alcoholism will obviously ultimately, and decompensation, which is really loss of function of that organ. Alongside that, there is a link with metabolic syndrome, so with diabetes and obesity. All of these things impact on liver disease progression. To some extent, the liver can regenerate, but there does come a point where you cannot reverse the damage that's been done, and the scarring is so significant and extensive that the organ is no longer viable. And when that time comes, the only option in terms of survival is to have an organ transplantation, a liver transplant. Many barriers to transplantation, demand far outstrips supply, and you have to go on lifelong immunosuppression. So it really is a insufficient standard of care to protect the patient. This slide just overviews how serious liver disease is as a health issue. And what you can observe is that unlike a lot of diseases, liver disease is getting worse. The mortality is increasing. And really it's now the third biggest cause of premature mortality, with a huge amount of years of working life lost every year. And as I alluded to earlier, that demand for liver transplantation is increasing year on year, and all these patients are on long-term dialysis. The quality of life is terrible, and a lot of them ultimately don't get a liver transplant, and effectively they die. So there's a real need and a narrative to develop treatments that can overcome liver disease and offer really an alternative to transplantation in that medical scenario. Going from the UK national to the global picture, again, these slides and the graphics illustrated within them really do just emphasize what are the causes of leading liver disease transplantation, and the fact that in Europe in particular there are a huge amount of deaths every year. And the disease causation, it varies in different territories, and I've just included this graphic of Japan just to provide some context. And in this respect, you can see that the patient stratification is slightly different in Japan than it is relative to Europe, for example. But overall, one can say that liver disease is a large and increasing global healthcare burden that really does have to be addressed with new technology. So what are the current options in terms of the medical management of liver failure? And there are two definitions that you should be aware of. You've got ALF, which is acute liver failure, which is really for people who've taken paracetamol overdoses and require emergency treatment. And there is ACLF, which is acute on chronic liver failure. For example, alcoholics, long-term damage to the liver, and eventually there comes a point where they require emergency treatment, so slightly different medical indications. But as you can see, for both of these indications, the treatment is really poor, substandard, and it doesn't give those patients any particular quality of life. With acute liver failure, you can have an organ system support, you have to stay in intensive care, dialysis, lots of drugs, really emergency leveraged type drugs. But the prognosis is terrible, frankly, and so there really does need to be a new paradigm in healthcare to assist the patients. With acute on chronic, where they've got long-term damage to their bodies, again, you tend to be given anti-inflammatory steroids to control and dampen the inflammation, and then there comes a point where you are medically scored against set criterion, and that can put you on the transplant list. So for those sets of patients who perhaps might be alcoholics, you know that they are on a pathway where they may eventually, once their symptoms become so severe and their bodies can no longer compensate, that they require organ transplant. So I've outlined this serious issue and global healthcare problem, liver disease. What's the solution? Well, several years ago, there was an inspirational, compassionate clinical study which was carried out at King's College by Neil Darwin. And basically, they had lots of very young children who really had early onset genetic liver diseases, and these kids had a terrible prognosis. And this particular group, they used primary human donor hepatocytes, encapsulated them in alginate, which is an inert 3D material, and inserted them into those infants' bodies in the perinatal fat pad. Amazingly, liver function was restored, and it allowed the withdrawal of the other types of medical support that was alluded to in the previous slides. And actually, in some cases, those small numbers of liver cells transplanted into the human body were shown to provide an effective bridge until transplantation for many months. So what you had here was a truly inspiring proof of concept human hepatocyte study showing that these encapsulated alginate beads, if you like, could restore functions in humans. But there's a major issue. You cannot get human hepatocytes. They're really scarce offshoots of surgical procedures. So they're not a scalable therapy for adults. However, the context and structure and demonstration of restoration of liver function will enable a solution to be applied using stem cells, and that's what we focus on. So just to give you a bit more detail on the compassionate study that was conducted at King's, there were 8 patients, and using the encapsulated human hepatocytes, 4 of those patients, effectively the treatment bridged them so they could make a native recovery. 3 of those patients kept those young children alive, so a liver transplant could be undertaken, and they survived, and 1 patient care was withdrawn and died. So, which is obviously tragic, but overall you can see that this therapy offered for the first time really, the potential to treat humans using liver cell hepatocytes encased in alginate, and really we have taken inspiration from that within Aculiv Therapeutics, and that's the focus of the remainder of the talk. So, having proof of principle that hepatocytes can regenerate function when placed inside the human body, then the next question is, what is the solution in terms of producing a medicine that's got a global reach that can treat the thousands of patients that would otherwise require liver transplant and those patients that have advanced liver disease? Stem cells are the solution, and in essence, Aculiv have developed a technology to allow a scalable source of high-quality hepatocytes to be generated on an industrial scale that can populate the liver therapy. And I should have pointed out earlier that the liver is unique. It's about 1 kilogram. It's one of the biggest organs in the human body. And you really do need massive volumes of cells, many billions of cells, or certainly high millions, to really introduce into the patient body and elicit a therapeutic response. And that is a huge challenge, and of course those hepatocytes have to be very high quality. So in terms of the medical product, we take stem cells generated in GMP conditions, use forward reprogramming, which is an incredible technology advance, to generate liver hepatocytes, and this is all done under GMP conditions optimized for cell therapy. They are then encapsulated in alginate, which is what one might term an immunoprivileged matrix. Crucially, at this juncture, that drug product or prototype drug product can be cryopreserved and stored for many years, and it can be distributed globally, which is obviously incredibly important. You can then distribute the medical product via standard logistics channels all over the world, and then at the hospital, the surgeon can, using what would be termed a low-grade surgical endeavour, use effectively a syringe to inject the medicine into the intraperitoneal fat pad of the patient. So that's really the therapeutic approach we're taking. So I should point out the team behind the project, and the research has been led by Professor Ludwig Valier. He was at the University of Cambridge for really about 15 years, and he's recently moved to Berlin. But to be clear, the technology was developed at the University of Cambridge. And Ludwig and myself co-founded Definigen, which is a leading liver cell company which works in the preclinical space, and it really provides a lot of high performance liver disease models, disease in a dish if you like, so that all the advanced therapeutic developers in the Boston areas and the key pharmaceutical companies can advance their liver therapeutics into the clinic in a rapid time. And so we do have a bit of a background in liver cell development, and that's obviously served as a foundation for the use of new technology in the cell therapy space. So what's the advantage of this technology? Well, really, when you assess this technology, it's based on two revolutionary advances. It's well known that when you introduce sets of transcription factors into stem cells, you can effectively direct their development and their lineage into different cell types, whether that cell type is neuronal, cardiomyocyte. However, there is a technical challenge, and you need an expression system that can drive the expression of those transcription factors in a really controlled way to really drive the performance. And that's what we've achieved here. Then, building on that, it's that combination of the inducible expression system with the sets of transcription factors and nuclear reprogram, which are, in one sense, controller mechanisms to really direct the transition of stem cells through key developmental pathways to produce very functional human hepatocyte cells. And the combination of those two approaches really enables a vastly amplified production of cells with a high degree of reproducibility in GMP conditions, production conditions that are suitable for cell therapy. So it's that combination of the expression system and the transcription factor and the reprogramming sets, which are novel in nature, that really drive this advance and the generation of billions of functional liver hepatocyte cells. So what's the future? Well, earlier I mentioned that there's an emerging area of drug development focused on developing therapies for NAFLD, which is non-alcoholic fatty liver disease, and it's a coming global healthcare threat. And clearly, we can develop therapies that can address this threat, restore function, and offer NAFLD patients a chance of an extended life with a higher quality of life. And alongside that, in terms of genetic liver diseases, there are really over 70 genetic liver diseases, many of which afflict children, sadly, which could, with the correct application of this therapy, be overcome. So you may or may not know background to this, but there are monogenic liver diseases. A single mutation causes a single enzymatic dysfunction, and that can lead to damage to the human body. So if we can develop therapies which target these orphan liver diseases, they only impact a small number of patients, but collectively, these are really very large treatment markets where a lot of patients are being damaged and are being by the disease, and these are very severe, early onset diseases, really poor quality of life, and unfortunately a lot of the children actually pass away before they reach adulthood. So there's a massive opportunity here to develop what I would term next generation therapeutics, increase life, transform lives, and offer some hope for the future for these patient sets. And I believe I've just about used up my time, so thank you again. Thank you.
Good, that's a video. Thank you very much for the patience and to stay with the line. And if you got questions to Marcus, or if you like, I put my email address, so he's happy to answer later on. And just one information, there's a big investment money coming into the Cambridge area for the stem cell of the world. And if you are doing similar research, etc., maybe why don't you collaborate? Good. All right. So two presentation has been finished, and if you can, Josefa is on, and Soyama-san and Takahashi-san. So we can start the panel discussion. So we will welcome Soyama-san and Takahashi-san. Soyama-san is CEO of LinkJ, and Takahashi-san is also in LinkJ, but having many years of experience in mega pharma doing open innovation. So before going to that, I got questions, so I want to prioritize. So first questions are both for Josefa, if you have read. So for the patient validation of your the original cell, would you choose the origin for scale up? Some are good for scale up, some are no. How do you select?
Yeah, so when we for both customers that are transferring their lines into Celeste for scale up, and also for Celeste scouting the world for potential lines that we can bring in to develop and then offer commercially to pharma and biotech. We look at, first of all, there's the market, what the market pull is right now for organoids and required for scale up. And at the moment, the market pull I'm seeing is for actually for normal and healthy organoids, such as colon, duodenum, so gastrointestinal organoids, and then also liver organoids, which kind of slightly aligns with what Marcus was saying in his presentation. So at the moment, I'm scouting for these lines around the world. And when we do, we basically have a roadmap to triage access. So first of all, we link up with key opinion leaders or those that have generated lines or published about these lines. Once we have connected with them, we in essence send them a questionnaire that asks them questions about the line they've generated, the what sort of what the growth conditions are, how long they've managed to passage this line on for. For example, if they say they've only managed to passage the line on for 2 or 3 passages, then we're unsure whether we can tech transfer this into Celeste and then develop it in our bioprocess, because we would want to be able to at least have a known passage of at least 5 to 10 passages to know that we can then continue the development in our hands. We want to know what sort of growth medias are required, what ECM is required. For example, at the moment, most organoids require, patient-derived organoids use Matrigel or BME as their hydrogel to ECM. That's great, but we would like to move away and use more defined synthetic hydrogels, especially as I see for the future, organoids not only being used in drug discovery and toxicology assays, but also in regenerative medicine. So actually, I've seen studies now where organoids have been used in transplantation studies. So if this is required, we need to move to being able to generate organoids at scale with GMP type conditions. So we need to move away from animal derived hydrogels. So in essence, when we're scouting for organoids, we in essence ask lots of questions about what that organoid is, how it grows, and in essence, see if it's compatible with our bioprocess and bioreactors. And then we look to tech transfer these lines into Celeste, either through an MTA or through some sort of sponsored research work And usually what I'm finding is that I cannot find organoid lines to transfer into Celeste because they do not have the appropriate ethics and commercial consents for future commercial use. When these organoids have been derived from the patient material, the consents have not been in place for commercial use. So that's the biggest problem I'm finding.
Sure, great, thank you. So quality, including gender-free and the ethics, so clear answer. Yes, thank you. Takahashi-san, maybe you have many questions.
Yes. So first of all, thank you very much for your wonderful presentation. And I felt the use of organoid in regenerative medicine and precision medicine is progressing. And like the PDX model, the PDOs will be commonly used. So here I have some questions. The similar services is available even in Japan. So could you explain a little bit more about, what makes your company unique? Or what kind of the technical advantage do you have comparing with other, the similar companies?
Yeah, sure. So we believe that Celeste is the only company in the world that has the technology to scale up the manufactured production of organoids. So there are other companies around the world that are generating organoids from tissues and perhaps manually growing these for small, small type, small compound number screening or other types of research studies. But where organoids might be required for high throughput, large volume applications, we believe Celeste is the only one that has a novel platform USP to, in essence, manufacture organoids at scale. So we take existing organoid lines that have been generated, whether in biotechs and pharma in-house, or whether they've been accessed through biobanks at the hub, ATCC, different cancer institutes around the world. We take these lines and we can bring them into Celeste, optimize them, develop them, and put them into our bioprocess and into our unique bioreactors, and then in essence scale up their production to large-scale batches, and then also be able to offer these large-scale batches on a regular occurrence. So we want to add value and longevity to these really good 3D models that have been generated from kindly donated patient donors.
That's good. That's good. I fully agree. Scaling up in the production is quite important to promote the further use of organoid in the new drug development or future clinical applications. Yeah. So the next question is the following: from the point of view of pharmaceutical companies, so pharmaceutical company would like to secure the free to operate, or they'd like to own the IP once they, successfully move on to the further clinical trial or the clinical application. So could you explain a little bit about the IP situation of your product or your services?
Yeah, sure. I'll try and explain it as I believe the IP. So there is an IP landscape around patient-derived organoids and their use of what the specific growth factors to grow these organoids. So that this is all part of the hub IP landscape and the hub organoid technology. So where those growth factors are required. So in my head, this is our spondin and Wnt for the growth of organoids. Then you require the use of hub organoid technology, and you require to speak to the hub about potential licensing, and they have various different licenses for different applications. So Celeste has the license to use hub organoid technology to scale up and manufacture organoids where we need to use their technology. So we do not offer, and that is our focus, is on the scale up and expansion of organoids and offering that as a service to customers and collaborators. How then those organoids are used in different types of applications, those users need to explore the IP landscape and decide what IP they require basically from that. Yeah. It's a very interesting ecosystem, and I think people need to basically look into this. But I also find, taking a step back, the other part of IP is that in essence, some researchers that are generating new organoid lines in their labs do not know that there's an IP landscape, and then when they come to tech transfer or license out their organoids, they find out actually, in essence, they are using someone else's IP already to generate that organoids. And I think, so I believe there needs to be a bigger, wide roundtable discussions around having everyone on the same page about what IP is being used to generate the organoids and what IP is required to then use those organoids in different types of applications.
Yeah.
And the next,
always.
One more question. Maybe, this question might be a quite mean question. So, sometimes I hear that the organoid culture is quite expensive because of the use of the cytokines and other, the reagents. So how about, is that true, or how about your case?
I think what that question, I think it is true, because obviously most people that think about growing cells think about 2D cell lines. So it's been 2D cell line and growing at scale has been going on for 50, 60 years, and it involves probably simple media formulation, simple plasticware, whereas growing organoids and other 3D models involves using also an ECM. It uses expensive, like you say, growth factors or specialized medias, and they all add to the cost of growing the organoids and then expanding them at scale. So what I find when I'm having discussions with some partners is that they're comparing to a 2D, they're comparing the cost to an existing 2D setup versus a 3D setup. And what they need to think about is actually the added value of testing compounds and novel chemical entities on 3D models that have actually been derived from patients, rather than a 2D line that has been growing for the last 50 years. And the data out there shows that there is no correlation between the 2D line hits versus animal models versus clinical trial results. In oncology, for example, only 5% of drugs get to market are successful. So that's a 95% failure rate. Is that because we are not using the right models earlier in drug discovery? And the right model in my head is those that have been derived directly from patients.
Yeah. Thank you.
Yeah, interesting. Always comparison between 3D, 2D as a value.
Yeah,
yes.
It's that question of cost versus value.
Yeah.
Great. Somehow, do you mind picking up a couple of questions from floor?
Sure.
Yes. The second question is a simple technical, like what is the benefit between patient-derived organoids versus 3D culture human cell line? And second question is how to maintain the immune cells in organoids in a bioreactor process.
Okay, so I'll do the second question first. So in terms of patient-derived organoids, these are adult stem cell derived from epithelial stem cells. So there are no other cells. They are basically those organoids that have formed from the adult stem cells are all from the epithelial, differentiate from epithelial cells. So there are no immune cells or vasculature or stromal cells. We are growing epithelial derived adult stem cells, and then we basically deliver these to customers who then are exploring, can they leverage these organoids with other cell types to create more complex in vitro assays? So combining with either fibroblasts or PBMCs, or in some cases combining with microvessels to create some microvasculature. So that's the second question. The first question was on the difference between organoids and 3D models. Was that right?
Sorry, I was speaking of other questions. Yes.
I was just
what is a patient-derived organoids versus 3D cell culture?
Yeah, in my head, a patient-derived organoid, as I said, is so taking a biopsy or resection from that tissue, isolating or growing the adult stem cells from that tissue in a 3D environment, so they never ever are in a 2D planar environment, and consistently in that 3D environment, and allowing those stem cells to differentiate and then culture on. This is different to iPSC derived organoids, where you're taking iPSC stem cells and then programming them into different types of cell types, and this is different to perhaps cancer stem cells or cancer cells or patient-derived cells, which are then grown into spheroids mechanically. So in terms of patient-derived organoids, this is taking the adult stem cell and then allowing the biology to form the 3D multicellular structures, in the right standardized and optimized conditions with the appropriate growth factors or ECM, basically.
Thank you. One more technical, sorry, sir, 1 minute. How to control the size on your bioreactor?
Yeah, so the way we do this is actually we believe our bioreactors help with size control. So in essence, when we come to seed our bioreactors, we're seeding the bioreactors in single cells. So they're all single cells in these bioreactors. And because we're basically providing a really consistent and reproducible environment where there's a continuous perfusion of media, we're ensuring that media has there's no issues with metabolites such as lactate levels, and there's high glucose levels and oxygen levels. That environment basically allows all those single cells to grow at the same rate, and to in essence, when we come to harvest, we have got a good population of size control organoids. We can then add, on our customers' request, some straining, so we can filter through straining to specific sizes, but we believe our bioreactors give us this control of size. And this is completely different to how organoids are grown manually, because they're grown in domes of matrigel. So you have differential effects of growth factors and media hitting, coming to the organoids that are different in the center of that dome to the outer edges of the dome, and therefore you end up with different size organoids. So our bioreactors enable us this size control. And obviously there's papers out there that show size correlates with viability. So if we can deliver good size organoids, they are viable. So this enables then our customers to get good quality data, which is shown by reducing in error bars, good Z prime values, and such, basically.
Interesting, thank you.
So
sorry.
quick questions. In terms of the the reproducibilities, so you've also confirmed the reproducibility among production batches, right?
Yeah, so this is our key element. Every time we produce a batch, we have a distinct QC package, call it QA package, to ensure that each batch is similar or the same. And also, because each batch is produced from the same a working stock, which is linked to a master stock, we will always produce a batch to the same passage number, and passage number is also linked to good quality organoids. So in academia, people are continuously passaging on their organoids. So over time, maybe this will be detrimental to that line, whereas we always know we're going back to a tiered bank stock of organoids, expanding these organoids, and then delivering these batches back to our customers, basically. Yeah. And the quality aspects. So in terms of QC, which is we are continuously evolving based on what our customers want and what we see in the science. So we do some basic health and safety quality control. So measure for mycoplasma, measure for virals and endotoxin to ensure that this is good quality for health and safety. And then we do STR to ensure that the line has not changed. We do some compound validation. So we'll test our batches against some known compounds, and we'll just compare IC50s against batches. So as this is especially important for cancer organoids. And then other types of assays, sort of more bespoke, but we're kind of bringing this in slowly, is the genomic study. So we'll do whole exome sequencing and RNA-seq. And this is just to ensure that our process, we have not changed the genetics of this line.
Yes, I like the exciting discussion. Soyama-san.
Okay, I'd like to make a short comment at the end. Yeah.
Sure, great. Yeah, I think we answered all of the questions. If not, my apologies. But good. All right. So further connections. So for the audiences, please use like questionnaire in the end. And we are happy to connect either to Josefa at Celes and Marcus at Accurite. So I mean like your liaison. Good, and thank you very much. And sorry for like 5 minutes over, but timekeeper. Back to Simon-san.
Okay, so regarding organoid, as we discussed today, there still exist many challenges, I guess. But on the other hand, there exists huge potential, right? So I hope we overcome those challenges by gathering our wisdom, efforts, and enthusiasm. Thank you very much.